Hey everyone, welcome back to class. So we were just talking about how DNA can be made more or less accessible to transcription. There's another way we can control transcription, and that is through transcription factors. So controlling gene regulation is combinatorial. We have these multiple layers of control elements, and one of those is using transcription factors. Which allows you to establish different patterns of gene expression in different cell types, because different types of cells are going to have different types of transcription factors available. So here you have this first cell type has some regulatory transcription factors. It has some general transcription factors, and that's going to control if this gene is being transcribed or not. This cell is going to have some other transcription factors where its gene is going to be turned on. This has a different set of transcription factors available, and its gene is not going to be transcribed. It's going to be turned off. So these different combinations of having different transcription factors resulting in making or not making a protein at a high level, making it at a low level, or not making it at all. So there's this wide range of efficiency for gene transcription. You can make it a low level, a high level, or off at these different levels, depending on what regulatory factors are there. By changing these regulatory factors, you change the regulation of that gene. Most regulatory transcription factors can be placed into a small number of categories based on a motif that they have, or a secondary structure that they have to their shape. Generally, those motifs are going to be DNA binding elements, which allows them to bind to the DNA and regulate the transcription of that gene. And there are other motifs that will be able to stabilize other regions of the protein separate from DNA binding. One motif is a helix-turn-helix. So you have a helix, a little turn, and another helix. So a helix, a turn, a helix. Helix, turn, helix. Okay, so you have two alpha helices separated by a little bend. One alpha helix is a recognition helix, which recognizes and binds to DNA. And the second helix is just for stability, to stabilize this overall configuration. Examples of these types of proteins would be the LAC repressor, the tryptophan repressor, and the CAP protein, all of which we talked about in our early videos. Another would be the zinc finger motif, where you have an alpha helix and two beta sheets, and these are all held together by cysteine interactions and histidine interactions with this zinc ion, which it's named for. Zinc fingers will protrude from the protein surface, so whatever your transcription factor is, this will be sticking out from it, and these are going to be points of contact between the protein and the DNA. Examples of this would be transcription factor for the 5S ribosomal RNA genes, or transcription factor 3A. Next is helix-loop-helix. So instead of just having a little turn here, you have a whole loop here. So you have a shorter helix, a loop, and a longer alpha helix. These generally have hydrophobic regions that are able to connect to polypeptides, which may be similar or different to each other. Next is a leucine zipper, and how similar this looks to a zipper. I love this one. So you have these little leucines sticking out from your proteins from its three-dimensional shape, and these leucines just kind of interact with each other. So you end up with these coiled segments of the protein. So the leucines will interact with each other and interlock, allowing your proteins to coil around each other. And the DNA binding will happen when you have these two alpha helices connect adjacent to the leucine zipper. A couple of transcription factors, CREBs and STATs. These are types of proteins that can be activated by phosphorylation. So their default state in your cell will be altered by phosphorylating them. So protein phosphorylation, we've seen this before a lot as a method to activate or deactivate a protein. So cyclic AMP, this camp here, is a second messenger we see a lot in cell signaling. So you've had some kind of signal, a growth factor or something, that now you have activated cyclic AMP, activating and stimulating protein kinase A. And that protein kinase A is going to start phosphorylating a lot of proteins. One of its targets is CREB. CREB is able to bind to DNA at these cyclic AMP response elements. But when CREB is phosphorylated, it is going to now interact with a protein called CBP, or CREB binding protein. And when these are bound together, when CREB is phosphorylated and bound to CBP, it starts causing histones to be acetylated. When those histones start being acetylated, they loosen up their grip on the DNA, so you start having these nucleosomes being unpacked, and that means they're just kind of looser, more diffuse. The transcription factors can get in and activate. It can reach the DNA now, and transcription will be activated. So growth factor results in CREB being phosphorylated, interacting with CBP, which loosens up your DNA from that histone, and you turn on that gene. Another target of phosphorylation is STATs, which is another family of transcription factors. STATs especially are important in signaling after the reception of interferons. When an animal cell is infected with the virus, an interferon is a protein signal that it sends out to allow the cells around it to adjust. So when a cell gets this signal of an interferon, it knows that there is a virus nearby. It needs to activate genes to protect itself. So the STATs are going to be a way for the cell to activate those genes to protect itself from the viral infection. So when the interferon binds to the cell surface, it causes the receptor to cause activation of the protein JAK, or Janus kinase. The activated JAK is going to phosphorylate your STAT proteins in the cytoplasm, which causes them to dimerize. See how these phosphorylated ones are sticking together in pairs? And that'll go into the nucleus, bind to DNA, repair response elements, and turn on all of these genes, which are going to be responsible for allowing the cell to survive and proliferate and not be harmed by any viruses. If the virus gets into that cell, it'll be prepared with these genes turned on to shut down that viral proliferation. Okay, so we still have a few more methods of genetic control we're going to talk about. We're going to talk about how animals are able to control the creation of different body parts based on differential gene regulation, and how we can control our genes and our proteins downstream from transcription. All right, I'll see you then. Bye.